Hey guys, we are Group 2 and our theme is immunological memory. First, I'd like to start with what is immunity. Immunity literally means the system which gets away from plagues. To be more specific, immunity is a series of mechanisms that detect strange objects such as pathogens and cancer cells and eliminates them. This process is really significant in that we keep our health. I think Group 1 explained it for us, but just for a reminder, there are two types of immunity. One is innate immunity and the other is adaptive immunity. Innate immunity is the immunity we were born with. The innate immune system serves as the body's first line of defense, and it can react quickly to a wide range of germs. On the other hand, adaptive immunity, which we get after birth, reacts to specific germs. Now let me explain the subject of this presentation. Immunological memory. When cells divide in the immune response, some cells become memory cells. Immunological memory is a mechanism by which memory cells are acquired, and when infected once again, they react quickly and release antibodies. So how do memory cells actually work? At the first infection, B cells divide and produce antibodies, but at the same time, memory B cells are also produced. Memory cells quickly produce antibodies when infected with the same antigen for the second time. Thanks to this function, after the second time, infections do not occur or are mild. The roles of immune memory are mainly played by memory B cells and memory T cells. As seen in graph 1, antibodies produced by B cells remain in the body in small amounts, even after the infection is terminated. This is to prepare the body for an immune response the next time the same pathogen invades. You can see that during the second infection, antibodies in effector T cells proliferate more rapidly than during the first infection. How is such rapid immune response possible? First, let's look at the function of memory B cells. In the early stages of infection, naive B cells recognize the antigen with IgM receptors and become plasma cells that produce antibodies specific for this antigen. However, these plasma cells produce only IgM antibodies with low affinity for the antigen. In the late stages of infection, IgM antibodies, which have a higher affinity for the antigen than IgM antibodies, begin to be produced. Naive B cells with IgM receptors bind to IgG antibodies and undergo apoptosis, so this number of naive B cells with IgM receptors decreases as the production of IgG antibodies increases. Therefore, after the end of infection, B cells that produce IgG antibodies with high affinity for antibodies remain as memory B cells. In other words, when the same pathogen invades, these memory B cells differentiate and produce antibodies specific for the antigen much more quickly than during the first infection. Next, let's look at the function of memory T cells. First, naive T cells are activated upon recognition of pathogen antigens and begin to proliferate. During this proliferation process, naive T cells divide asymmetrically into effector T cells and memory T cells. Effector T cells are active in response to pathogen proliferation and are very short-lived due to the high stress on the cell. Memory T cells, on the other hand, divide slowly and are carefully maintained. Thus, they are quite long-lived and remain in the body after infection ends. Like memory B cells, these cells proliferate rapidly during the second infection, triggering a strong immune response that prevents infection. Lastly, I would like to explain the mechanism of immunological memory at gene level. The DNA of human genetic information is wrapped around a protein called histone so that they do not become entangled with each other and are contained in a cluster of histones. This state is called chromatin. In this state, genetic information cannot be read immediately, so it cannot be active when needed. However, when we get infected, chromatin is entangled and the macrophages are activated. This situation is maintained for a long time, so this is called immunological memory. Yeah, so I hope you guys understood what immunological memory is. This is the end. Thanks for listening.